Yes, so my name is Tony, and I'm happy to talk to you today about TRIM55 and its association with hypertrophic cardiomyopathy. Um, yeah, okay. So hypertrophic cardiomyopathy, short HCM, is the most common human inherited cardiomyopathy with an estimated prevalence of 1 to 200 in the general population. HCM is mainly characterized by thickened left ventricle, as well as myocardial disarray, increased interstitial fibrosis, and diastolic dysfunction. HCM patients can either show no symptoms at all, develop heart failure, or even die of sudden cardiac death. HCM is considered a sarcomyopathy with more than 1,400 mutations in genes encoding sarcomere proteins, and these compromise cellular homeostasis. Despite that HCM is this common, there's no curative treatment available to date, so there's a very high need to identify novel treatment options. Sorry. Yeah, so in the heart, it's very important to have the cellular homeostasis, and therefore we need the protein quality control so that the heart can work properly. If we have a healthy heart, we have a healthy cardiomyocyte that shows a functional sarcomere and normal protein turnover. The sarcomere is the smallest functional unit of cardiomyocytes that is responsible for the beating of the heart. To visualize this for you, here at the bottom, an immunofluorescence image of a healthy iPSC cardiomyocyte, where I stain for the sarcomere by staining for alpha-actinin 2, or short ACTN2, which is a marker of the Z disk, shown in pink, in combination with the ambient marker obscurin, shown in yellow. If we now have a diseased HCM heart, we have diseased cardiomyocytes with an impaired sarcomere and impaired protein degradation, either because the proteins can't be properly marked for degradation, or the degradation itself is impaired. To also visualize this for you, again, another immunofluorescence image here at the bottom, where you can see a highly disorganized sarcomere in a diseased iPSC cardiomyocyte. The protein I'm interested in is called TRIM55, and it's part of the ubiquitin proteasome system, which is the major proteolytic system within cells, ensuring cellular homeostasis. Here, TRIM55 is crucial as it is an E3 ubiquitin ligase and thereby ensures the proper transfer of ubiquitin onto the protein targeted for degradation. Multiple rounds of this lead to the polyubiquitination of the targeted protein and its subsequent degradation via the proteasome. TRIM55 is highly expressed in skeletal and heart muscle, and it has been shown to localize to microtubuli, to the ambient of the sarcomere, and to even interact with sarcomeric proteins such as titin. Due to its localization and interaction, it is assumed that TRIM55 plays a regulatory role of sarcomere formation. To date, various mutations in TRIM55 have been identified and associated with HCM, but the underlying disease mechanism is still unknown. However, we are in the very lucky position to have access to patient material from HCM patients carrying TRIM55 mutations. Our aim is now to identify the underlying disease mechanisms of TRIM55-associated HCM. And here you can actually see our cardiac disease modeling workflow, like how I want to investigate the underlying disease mechanisms. So therefore, I culture iPSCs, then I differentiate them into cardiomyocytes over a period of 15 days. Our differentiation protocol does not only generate cardiomyocytes, but also cardiac fibroblast, endothelial cells, and smooth muscle cells. I then analyze the cells either in a 2D cardiac monolayer or in a 3D human vascularized cardiac organoid. If I do analyze them in a 2D cardiac monolayer, I mainly do this to perform image-based analysis as well as molecular biology analysis, such as mass spectrometry. If I'm making 3D cardiac organoids, we actually do this in a 96 well plate format. And here in the video, you can see, so those cardiac organoids, they form around poles. And then if they're properly developed, they move the poles. And this allows us to analyze the contractility of our cardiac organoids. So here you can actually see a scheme of the TRIM55 gene and the corresponding protein with its important domains. To now assess its putative disease-causing properties, we decided to generate a TRIM55 knockout line, and therefore we're targeting the first two exons of TRIM55 by CRISPR-Cas9 gene editing, aiming to prevent overall gene expression. After the successful generation of this line, we first validated the levels of TRIM55 protein by immunofluorescent staining, as shown here on the left-hand side, as well as by immunoblotting, shown here on the right-hand side. Both methods revealed a clear absence of TRIM55 protein. After the validation of the TRIM55 knockout line, we actually were very interested in the functional phenotype of these cells. Therefore, we decided to look into calcium imaging, as an abnormal calcium handling is actually a disease hallmark of HCM. So therefore, we decided to assess the calcium handling in our health control and in TRIM55 knockout iPSC cardiomyocytes using fluor4AM, which is a cell-permanent calcium indicator that exhibits an increase in fluorescence upon calcium binding. And actually, you can see this here in the video on the right-hand side, where you can see the release of calcium in form of a fluorescent spark in between contractions. And then this analysis actually showed that our TRIM55 knockout iPSC cardiomyocytes have a markedly prolonged calcium transient in comparison to the healthy control, which you can see in those two graphs. The knockout is shown in this purple pinkish color and the control in the mint color. So this really speaks for abnormal calcium handling. What we also saw in our cells was a lower beating rate as well as a longer beat-to-beat distance. Next, we actually made 3D cardiac organoids of our healthy control and TRIM55 knockout iPSC cardiomyocytes and assessed contractility in this format. And this showed that the TRIM55 knockouts have a higher force, a lower beating rate, a prolonged activation, and relaxation time in comparison to healthy control. And these results are quite promising as they hint towards an HCM-like disease phenotype. So for now, I already talked a lot about the functional phenotype, but we also wanted to figure out what is more the underlying causes. So we thought, let's do some proteomics. So we did some total proteomics analysis of our healthy control and TRIM55 knockout. And here on the left-hand side, you can see the volcano plot, which confirmed the reduction in TRIM55, shown in purple, as well as an increase in TRIM63, shown in yellow. And this is quite interesting. TRIM63 is a very close family member of TRIM55, and they are known that they can compensate for each other. Here on the right-hand side, you can just see the Venn diagram of this data set, which shows that we had significantly dysregulated proteins that were either up or down regulated in our knockout in comparison to healthy control. Our bioinformatician Pooja then helped me and generated the gene set enrichment analysis of this data set, which is shown here. And this revealed, and we were kind of relieved to see that we have a marked reduction in muscle contraction, so in contractility in our knockouts, which emphasizes the role of TRIM55 in sarcomere formation or turnover. Further, we also see a marked decrease in the metabolism, but on the other hand, a marked increase in DNA replication and RNA processing. To follow up on this, we decided to also perform bulk RNA-seq of healthy control and TRIM55 knockout iPSC cardiomyocytes. And after first validating the dataset, Pooja also here generated the gene set enrichment analysis for me, which again showed a marked reduction in contractility in our knockouts. We also saw a marked reduction in metabolism, as well as protein degradation. And on the other hand, we saw a marked increase in collagen turnover and remodeling and ECM remodeling. So I already talked about a lot, like total proteome and RNA-seq, but then we were like, okay, TRIM55 plays such a crucial role in the ubiquitination cascade. So what about the cardiac ubiquitinome? So we decided to assess it in healthy control and in our knockouts. But why specifically the cardiac ubiquitinome? So because we know that ubiquitination, by ubiquitination, you can actually modify the function of a protein, either by degrading it, but that's not the only way how it does it. So it can also modify complex compositions of proteins or the localization of a protein or its activity. And we do think that, especially in the knockouts, as TRIM55 plays such a crucial role in the ubiquitination cascade, that we will see differences in the ubiquitination profile. Here on the right-hand side, you can actually see our workflow. So we culture healthy and TRIM55 knockout iPSC cardiomyocytes, lyse them, digest them, purify the protein, and then basically the most important step is that we perform an IP using diglide beads with a KEG antibody motif. This is commercially available for cell signaling, and this allows us to pull down all the ubiquitinated peptides in our lyse it, and then we validate our work on a Western blot, and then we run the samples on the mass spec. And for us, the biggest bottleneck really was the amount of protein we needed, because only 1% of all the proteins are ubiquitinated. So we needed a lot of protein and cardiomyocytes for this. But it seemed to have worked. So here on the left-hand side, you can see the Venn diagram. Just to flag it, if you analyze the ubiquitinome, you always run the total proteome as well as the ubiquitinated peptides. And this is shown here. So we identified around 8,400 total proteins and around 14,300 ubiquitinated peptides that correspond to 3,996 genes. So this was a really good run. So as around 94% of all ubiquitinated proteins were also identified in the total proteome. And then in this run, we actually found 515 significantly dysregulated ubiquitinated peptides, which we thought, oh, that's very exciting. So we definitely do have a difference. And then we tried to take a closer look. And then I needed the help of Pooja again, because I mentioned, so you do the total proteome and then your ubiquitinated peptides. But if you really want to see the effect, or if the ubiquitination is really lower or higher, you need to normalize the ubiquitinated peptides to the total proteome. So this volcano plot is showing the normalized ubiquitinated peptides. And the first thing we checked what about the TRIMs? So TRIM55 is shown in purple, markedly down. And then we also checked its two closest family members. So there's TRIM54 in mint and then TRIM63 in yellow. So both of its two close family members seem to have a lower ubiquitination in the knockouts. So I first want to go a bit deeper into TRIM63. So as I said, we saw significantly lower ubiquitination at 11 sites. And then if I had a closer look at where are those sites, one of them is directly at the N-terminus. And I forgot to mention, I used the visualization tool for this. It's called SCV Lab. And I used the flanking sites of the case that were ubiquitinated. So yeah, so one was at the N-terminus, whereas other three were located within the coil domain. So this is where the TRIMs, so if TRIM55 and TRIM63 can actually form homo or heterodimers. We also identified some within the titin interacting domain, and this is very interesting to us, as it has been shown before that titin does interact with TRIM55 and TRIM63. However, we also found significantly lower ubiquitination within the cos domain. The cos domain is important for the interaction with the microtubuli, and we found a significantly lower ubiquitination within the ring domain. And this is also very interesting, as the ring domain, it's where the TRIM63 actually interacts. So we're going back to the ubiquitination cascade. This is where the interaction with the E2 ubiquitin conjugating enzyme takes place. So we thought like, oh, this looks interesting. So let's have a look at TRIM54. And that's what you see here. I used the same visualization tool. And for TRIM54, we identified 4 significantly lower ubiquitinated sites. And we found one again at the N-terminus of TRIM54, one within the cos domain. So, and then, so the cos domain and also the microtubulin homo-oligomerization, domain, where the TRIMs again can interact with each other or with the microtubuli, and then one within the B-box domain. And this is where TRIM54 can interact also with other proteins. So this looks quite promising. We're still working on trying to understand what that means. I mentioned the interaction with titin before. So we actually looked at titin. As you all probably know, it's the largest human protein. And you can actually see it here on the left-hand side. In our total proteome, we actually detected around 60% of it all over the protein. So the detection was really good. And then we actually detected a lot of ubiquitination sites, 649. I think so far there were only around 250 published. So I think we had a very good and deep read. So yeah, again, I just visualized the flanking sites, but they were really all over, tightening equally distributed. But then I had a look like how many of those were actually significantly dysregulated. So this is what you see here on the left-hand side. We found 125. And out of those, 55 showed a significantly lower ubiquitination and 69 a significantly higher ubiquitination. So this is obviously a big number. So we were first like, okay, there are a lot in IgG-like domains. So what does it mean? But we were actually quite excited that one of those is actually within the protein kinase domain of titin, and this side shows a higher ubiquitination. So currently we're still working on what does this mean. Yes, we're trying our best, but as I said, titin is the largest human protein, so it's not that easy to tackle, but we're working on that. So now we looked at the total proteome, the transcriptome, the ubiquitinome, and then we were like, okay, what about the interactome? So here on the left-hand side, you actually see our workflow. So we culture healthy iPSC cardiomyocytes, and then we lyse the cells. And in this lyse, there should be a mix of TRIM55 bound to its interaction partners. And then we just did an antibody beats pull-down approach. So we incubated the cells either with an isogenic control antibody or with a TRIM55 antibody, pulled them down, validated it by Western blot, and then ran the samples on the mass spec. And then here on the right-hand side, you can see the corresponding volcano plot of this experiment. And then the first biggest question for us was, where's TRIM55? And yes, we highly enriched this. It's shown here in purple. But we also highly enriched TRIM54, shown in mint, and TRIM63, shown in yellow. And this has been published before, that they do interact with each other. So this seemed to have worked very nicely. But as you can see, there's also a few other proteins that came up. Some of them we also identified before in other studies, like APK3. So yes. Okay, so I talked now a lot about the TRIM55 knockout and all the omics data sets. But I mentioned in the beginning that there's patients that actually do have a mutation in TRIM55. So I want to go back to that. And then I have a patient that has a deletion mutation, the C312 to 314 del cut. And this mutation is located within close proximity to the ring domain. And the ring domain is, I think I mentioned it before, just to highlight it again, it's where TRIM55 interacts with the E2 ubiquitin conjugating enzyme. So it's very vital for the ubiquitination cascade. So here you can actually see clinical data of this patient with the del cut mutation. At the top, you see the ECG, and then at the bottom, MRI images. And this is basically supposed to show you, yes, the patient is sick, yes, the patient has hypertrophy. But this is still very preliminary data. I literally measured those cells 2 weeks ago, 3 weeks ago. So we have patient-derived iPSC cardiomyocytes from this patient. And we also generated corresponding isogenic control. And then I made 3D cardiac organoids from these cells. And this is the data you see here. And we detected a markedly higher force, a lower beating rate, a prolonged activation, and relaxation time in comparison to the isogenic healthy control. So this looks really promising that we can confirm the disease phenotype in our in vitro culture, but obviously we need to reproduce this. Okay, so this brings me to my summary. I generated a TRIM55 knockout line and made iPSC cardiomyocytes, and those seem to have a functional phenotype that resembles HCM, and this includes a prolonged calcium transient, prolonged relaxation time, and a distinct ubiquitination profile, and especially there seems to be a link to titin that we want to take a closer look at. We looked at the TRIM55 interactome, and this confirmed the interaction with TRIM54 and TRIM63. We are currently working on a second set of interactome where we use a different approach to just validate this, as we were also hoping to identify sarcomeric proteins. We do have a patient, an ATM patient, that carries a TRIM55 deletion mutation that presents clinical with hypertrophy, and the corresponding patient-derived TRIM55 deletion del cut iPSC cardiomyocytes show a dysfunctional phenotype, including a prolonged relaxation time. So where I'm at and what's planned in the near future. So right now I'm actually also working on the in vitro localization of TRIM55. because we really want to see where it is. Is it at the sarcomere and what is it doing? And therefore, we decided to generate a double reporter cell line. So we decided to tag TRIM55. So here again, you see this scheme of the TRIM55 gene. And then we decided to tag it at the C terminus with green lantern. And then we thought, okay, nice, now we can see TRIM55, but we would also like to visualize the sarcomere at the same time. And there I actually used a cell line that was recently generated in our lab by Alan and James, where they tagged ACTN2 with mScarlet. So this really allows me to see both at the same time, the sarcomere and TRIM55. And then actually here you see a video of the cell line. So in green, we see TRIM55 and in yellow ACTN2. I image the cells over 18 hours. And despite a certain degree of photobleaching in the green channel, you can actually see that TRIM55 is either at the embed, so we see a striated pattern, or dispersed all over the cell. And then if we see this dispersed all over the cell staining, we either see it in form of punctate or a rather diffused signal. If we now bleach TRIM55, we see that the signal for both TRIM55 and ACTN2 is gone immediately. But within 15 minutes, you can already see green dots for TRIM55. And then over the 18 hours time frame, the signal for both TRIM55 and ACTN2 is coming back, but to a lower extent. Okay, so this is ongoing. Then I'm also working on a 2D disease modeling pipeline. So I want to evaluate further disease hallmarks of HCM, such as hypertrophy. So I'm looking at cell area and I'm looking at the number of nuclei. I look at the sarcomere organization, and then I also look at contractility. And actually, I went last week to Nanion and worked with Marta together, and we measured my cells on the cardiac side and also on the atlas, so we can have a look at the functional phenotype in 2D. And this seemed to have worked nicely, but we're currently still analyzing the data, but it's looking good. And then I'm also, by now, have a full suite of cell lines. So I have patient-derived, but I also have healthy cell lines where I introduced mutations that I'm working with, so patient-derived and then mutated iPSC cardiomyocytes, and I just showed you the first preliminary data of this. I would like to conclude by acknowledging my lab, the Heart Regeneration and Disease Lab, as well as all of our collaborators, and I would like to thank you for your attention, and I'm happy to take your questions.
Thank you, Antonia. It was a really great talk. Really full of information and full of data. Such a great story. I have a question for you. So you, of course, mentioned that you have patients with this specific mutation of TRIM55. And do you think that actually different mutation will give you a different phenotype? Or have you any data supporting that?
I don't have the data yet, but I'm assuming so. So what we see when we look in HCM in general, or in human inherited cardiomyopathies, for instance, like I think it's very well described for C protein or MYH7, that depending on where the mutation is, in which domain, it can actually lead to HCM or DCM. And then we either have a phenotype that is more associated with a loss of function, so the protein is not functional, or we have a poisonous peptide effect. So this in the end leads to a very different, so some have hypercontractility, some have hypo. So I do think that makes a difference. That's why I also looked at where the TRIM55 mutations are. We do have another patient that I will start with the cell lines. I will start working with in a few weeks, where it's in a different domain within the B-box domain, which is important for protein-protein interaction. So I'm curious whether that cell line has a similar phenotype or not. I just know from the clinical data that both patients have a very similar phenotype. We're currently trying to find more patients, but it's a very rare, TRIM55 mutation, that's quite rare, but yeah.
Okay, thanks a lot. There is a question now from the Q&A. So the first question is that TRIM54 is an emerging drug target in the hepatocellular carcinoma. Are you planning to execute functional investigation in hepatocyte cells?
We haven't. I saw those papers. I know it's also that I think it's lower, and if they reintroduce it, it improves. So it's lower, and that's why tumor development, if I got it right, and that they thinking of reintroducing it to hold or stopping the tumor development. I haven't really thought about that. I think in general, there's not a lot known about TRIM55 in general. There's a bit more known in heart and skeletal muscle because that's where it's mainly expressed. But I feel like it's more and more coming up also in different, especially tumors. So there's the hepatocell carcinoma you talked about, but there's also a study that showed its association with apoptosis in a different tumor, which I just forgot how it's called, but I think it's more and more coming up, and I think it's definitely an interesting target. So why is it so important for tumor development? I think it's because it's also a transcription factor, but I haven't really planned anything yet.
Okay, thanks a lot. And then I think there is another one. Okay, so she's thanking you for the nice talk, first of all. And then since the contraction parameters are finally affected by many proteins responsible of the release and reuptake of calcium from the sarcoplasmic reticulum, have you checked whether they are altered in the TRIM55 derived cardiomyocytes model? That's the first question.
I haven't. Very good question. We're currently always discussing what drugs we could add. Do we want to block the SR? What do we want to do to figure that one out? But I haven't taken a look yet.
Okay. And she has a second question. So how do you calibrate calcium concentration for amplitude quantification in concentration traces? And then involved in, it's a missing question, I guess. But how do you calibrate calcium concentration for amplitude quantification in contraction traces?
I just, so I'm not, so we're working currently on a image-based analysis, which allows me, if I'm using the a tagged line, to also assess contractility at the same time. But at the moment, I'm just doing calcium measurements using Caltrack. It's a published protocol. And so I'm basically just adding the dye, and then I image them, and I do image-based analysis of the calcium burst. And that also gives me information about the contractility. That's true. But I can't paste them or anything. So I'm very limited still. So we're working on improving at the setup.
Okay, thanks. And maybe just to conclude, since we have 3 minutes available, do you think that the other TRIM, so TRIM what were 63 and 54, would it lead to the same phenotype that you're seeing now?
It's a very good question. Actually, both are associated. No, maybe I'm wrong. TRIM63 for sure is also associated with HCM. There's also patients that have been identified, and they seem to have a similar phenotype. There is not so much done in the mice, because the problem is in mice, it's only if you knock out both, or if there's a mutation in both, then you see cardiac phenotype. So it seems to be that in human, this works differently than in mice. So there's not that much done in mice. But yes, there's definitely patients that have very similar proteins, so phenotypes. So we are thinking about making also TRIM63 lines and TRIM54.